Hello, my name is Yan Meng. I am a second-year PhD student at the School of Life Sciences of the University of Nottingham. This presentation is about adipose depot dependency of chloride channels expression in murine white fat. Firstly, what are white-fat adipocytes? Adipocytes, also known as lipocytes and fat cells, are the cells that primarily compose adipose tissue, specialized in storing energy as fat. Adipocytes are split into two main types, white and brown, that store and burn energy respectively. In white fat adipocytes, a large lipid droplet occupies more than 85% of the cell volume, squeezing the cytoplasm to the edge of the cell. It has been reported that the passive diffusion of chloride was found to predominantly regulate the resting membrane potential of white fat adipocytes, demonstrating the role of the chloride channels in preserving white fat adipocytes' normal physiological activity. On this basis, our aim was to identify these chloride channels at the molecular level. Both primary WFA and 33L1 cells were used for this study. For primary WFA, male SD rats of weight 200 to 420 grams were used to isolate epididymal, mesenteric, pararenal, and subcutaneous WFA. The locations of these fat depots are shown as the figure. Also, subcutaneous WFA were isolated from male BALBC mice of weight around 20 grams. Figure 1 displays typical pictures of undifferentiated and differentiated 33L1 cells stained with oil red O and hematoxylin counterstain. The accumulation of lipid droplets characterizes the differentiation of 33L1 cells to adipocytes, which can be seen in certain cells with or without staining. As fat accumulates in the cells will become less flat and more rounded. Mature adipocytes are fully differentiated and contain a single coalesced fat droplet. RNA sequencing was used to examine the expression of plasma membrane chloride channel-related genes in rat epididymal WFA. It can be seen that the chloride channels ANO1, LRRC8A, TTYH2, and 3 are expressed in REWFA, as well as LRRC8B, C, and D, the non-essential components of the volume-regulated ion channel. LRRC8E, another non-essential component of the LRRC8 family, and TTYH1, one of the Tweety family members, are hardly expressed in REWFA. The qPCR results show that the chloride channels have different expression patterns among different depots. For chloride channels in different rat WFA, LRRC8A showed significantly higher expression levels than the other chloride channels, while in mouse WFA, TTYH2 or 3 showed higher expression levels than those in rats. ANO1 was the lowest expressed chloride channel. When it comes to the LRRC8 family members, LRRC8A and C mostly expressed higher than B and D in different WFA. The comparison of relative expression levels of the chloride channels among depots show that different fat depots have different expression levels of these chloride channels. However, for LRRC8D and ANO1, no significant expression difference was detected among different WFA. Comparison between the undifferentiated and differentiated 33L1 cells showed that LRRC8C and TTYH3 were expressed significantly greater in undifferentiated 33. In conclusion, this study provides direct corroborative evidence for the existence of chloride channels, LRRC8A, TTYH2, 3, and ANO1 in WFA. These chloride channels express at different levels and show various expression patterns among different WFA depots and 33L1 cells. LRRC8A and TTYH3 appear to express the most. The putative properties of these chloride channels in WFA can be systematically addressed with further molecular evidence, and electrophysiological exploration for a better understanding of the roles in the physiological functions of WFA. Thank you.